Hello, thanks to everyone for joining us for this webinar, which is part of the Institute for Carbon Removal Law and Policy's ongoing webinar series on carbon dioxide removal issues. My name is Will Burns, and I'm the co-director of the Institute. The Institute is a research center at American University in Washington, D.C. that focuses on issues of science, law, policy, ethics, and public engagement associated with emerging carbon removal approaches. In the past few years, there's been extensive discussion of the potential role of agriculture in enhancing uptake of carbon dioxide through so-called regenerative farming practices. In today's webinar, two experts from the Innovative Genomics Institute, a research consortium of the University of California at Berkeley and the University of California at San Francisco, will discuss the potential role of genomic editing in potentially increasing the carbon uptake of crops, as well as generating a host of co-benefits. And so at the outset, I'd like to introduce our two speakers from IGI. First is Dr. Brad Ringeisen, who is a physical chemist who holds a PhD from the University of Wisconsin at Madison and is a pioneer in the field of live cell printing. Prior to joining the IGI, Dr. Ringeisen served as director of the Biological Technologies Office at the U.S. Defense Advanced Research Products Agency, or DARPA. Our second speaker will be Dr. Melinda Kliegman. Dr. Kliegman holds a PhD from Stanford University in the field of biology. Prior to joining IGI, she worked at the Bill and Melinda Gates Foundation, where she was a fellow in global policy and advocacy, advising on the use of genome editing in agriculture and human health. In an earlier role at the U.S. Department of Agriculture, Melinda served as a science advisor to the Foreign Agricultural Service. The structure of today's webinar will be interventions by each of the respective speakers, followed by Q&A with our audience. So I encourage audience members to begin to populate the Q&A section with questions as they arise. And with that, I will now yield the floor to Dr. Ringeisen.
Thank you so much, Will. And I'd like to thank the Institute for the invitation to speak today as well. Melinda and I are both really excited to be here to discuss some of the new things that the new research efforts that the Innovative Genomics Institute is trying to bring to bear to solve probably what's the largest issue facing society right now, which is the climate crisis. And you don't have to look much further than the news. It seems like it gets worse every summer. It's happening again this year with floods and fires. And, you know, this is something that we're tremendously passionate about at the IGI. And look, I was at DARPA. It's one of the preeminent research organizations in the government, the Department of Defense. But I really had a difficult time funding all of this amazing work in health and in sort of supporting the warfighter when I knew what biology could potentially do to help with the climate crisis. And so when Jennifer Doudna interviewed me for this position about a year ago, I decided now was the time that genomic technologies and bacteria and plants and crops have the ability to help solve and not be part of the problem, but to help solve the climate crisis. And that's really what Melinda and I are here to talk about today. So with that, I'm excited to share with you, if you go to the next slide, I'm excited to share with you the IGI mission. It's to bridge revolutionary genomic and genome editing tools. That's the CRISPR-Cas molecules and next generation molecules that you all hear about in the news and with the recent Nobel Prize, and to bridge those tool development to affordable and accessible solutions in climate and health. We have focus areas in sustainable agriculture and scalable climate solutions, but we also do a tremendous amount of work on the health side with CRISPR-based cures, therapeutics, and diagnostics, as well as developing the next generation of gene editing tools. It doesn't just stop with CRISPR-Cas. There's a tremendous new library now of tools that help researchers go in and essentially edit and change and manipulate, replace genes, regulate genes, turn genes on, turn genes off. There really is just a tremendous amount of new research going on, both in our institute and across the world. But one of the things that makes the IGI tremendously unique is that when Jennifer founded this institute, her goal was to be able to achieve equitable and affordable access to these genomic innovations. She truly believes in her core that CRISPR-Cas and gene editing can help bring a better life to all those in the world, that it's not just for those that can afford it, that there are equitable and affordable access that these genomic technologies and genomic innovations can provide. So next slide. So who is the IGI? I've mentioned Jennifer a couple times. Jennifer Doudna, the winner of the 2020 Nobel Prize in Chemistry for the discovery of CRISPR-Cas9. She is our founder and president of our governance board. Then there's myself as the executive director, and Mike Botchin, the dean of biology at UC Berkeley, as the deputy director. But we also have leaders in human health and sort of advancing genome engineering, as well as climate and sustainable agriculture. The list of names here: Alex Marson, Fyodor Ernov, Jill Banfield, Ross Wilson, Brian Statskowitz. These are all stars unto themselves. I'm really honored to be able to work with each and every one of these people on a daily basis. And not to mention Melinda, who I feel lucky to have stolen from the Gates Foundation about 6 months ago. And so this is the leadership team of the IGI. And we all are focusing towards that goal to both bring new cures to those that need them affordably, but also to be able to bring genomic innovations in agriculture, as well as soil and the environment, to be able to help solve this climate crisis that we're all experiencing. All right, next slide. So here's a slide that pretty much outlines what the IGI is thinking about when we say our climate strategy. Its foundation is in living organisms. These are organisms who have evolved over billions of years to be able to use all of the greenhouse gases that you all hear about. Not only CO2, carbon dioxide, but also methane, also nitrous oxide. Those are the top 3 greenhouse gases. They're biological organisms and biological mechanisms that can either uptake those gases or it can release those gases back into the environment. And so because that's true, we believe that gene editing and these organisms can be used to be able to manipulate those organisms to be able to soak up more carbon, to be able to direct the flow of carbon deeper into the soil, deeper into environments, to be able to store that carbon safely, not just safely, but actually to the benefit of farmers and to the benefit of the environment, because you could make more nutrient-rich soil that doesn't release greenhouse gases, but captures those greenhouse gases and is able to increase the fertility and increase the yield of agricultural crops, not to mention also potentially providing the economics of scale for carbon credits and such as well, okay? So we have centered this around 2 projects with the goals of reducing, creating solutions to minimize greenhouse gas emissions. Again, those greenhouse gas emissions now, at least in natural settings, like agricultural lands, are produced by living organisms, mostly living microbes and fungi. And we believe we can use those organisms to reduce those greenhouse gas emissions while maintaining food security. Restoring, that's what I was mentioning about the increasing the carbon content and the nitrogen content of soils. We believe we can build soil carbon storage back to pre-modern agricultural levels. We've been steadily depleting the carbon reserves in those soils, which hurts everyone. It hurts the environment, it hurts the farmer. We believe we can restore those levels. And then finally, revive. We think we can enhance crop yields with accessible and affordable climate-friendly strategies. Our first 2 projects in this are first, a pathway to the net zero farm, and that's focused on both reducing the farmer inputs. These are things like fertilization, pesticide use, irrigation. Each one of those comes with its own carbon footprint, and in the case of fertilizer, actually results in nitrous oxide emissions as well. And so you get a doubly bad environmental hit when you over-fertilize or when you use chemical fertilizers. Our second goal is to minimize those greenhouse gas emissions that are primarily coming from soil microorganisms, but they release both methane and nitrous oxide. And so that's the goal, is to understand the complexities of that system and to try to minimize the emissions of these harmful greenhouse gases from natural environments and agricultural lands. The second project is called Accelerating Biological Carbon Capture and Sequestration. I have a feeling that's probably the one that's most interesting to this community. But we really have three goals there. The first is to enhance carbon uptake via photosynthesis. Really, I'm not sure how much you all know about photosynthesis, but there's really two primary components of photosynthesis. There's the light reaction of photosynthesis, and then there's the carbon capture and carbon fixation mechanism that's centered around the molecule rubisco. We actually have partners on both sides of that house, and we think we can edit crops to both enhance that photosystem as well as enhance the carbon capture and carbon concentrating mechanisms in the plant to increase photosynthetic yields maybe to 20, 30, maybe even 50% over where they are today. That would increase both biomass above ground to increase yields, but also increase biomass below ground, which could help store and push more carbon into the soil. And the deeper you go in that soil, the longer you're able to keep that carbon stored deep in the soil. The second goal is to redirect the biological carbon cycling in soil to promote geologic storage. What you don't want with the biological biomass, like the plant biomass or soil microorganisms, when they die, for that carbon to be released back into the atmosphere. We think we can actually manipulate the pathways between the plant and the microbes to be able to encourage soil aggregation, to be able to encourage the types of microbes that utilize methane and utilize CO2 directly to be able to pump more carbon for longer periods of time into soil. And then third, there's actually an underutilized mechanism of carbon uptake that's actually non-photosynthetic microbes. Most people, when they think about biological carbon capture, focus solely on photosynthesis, whether that's algae in the ocean or seaweed, or whether it's photosynthesis from crops or grasslands. Those are all viable options, but there's an untapped resource that's sitting below ground. And those are non-photosynthetic organisms that live in dark environments. And they've evolved over time to actually utilize and uptake CO2. And they do so in some cases with even higher efficiencies than photosynthesis could ever achieve. So we actually have professors that are looking at those specific mechanisms and looking for ways to be able to enhance those organisms and enhance the mechanisms of that non-photosynthetic process. All right, next slide. So this is a summary of what we're talking about. The goal is to reduce those farmer inputs. That it reduces the carbon footprint of agricultural yields, which produce something on the order of about 25% of the of the overall emissions, anthropogenic emissions by human activity. That's goal number one, is to use microbiology and gene edited crops to be able to reduce those farmer inputs and also then reduce the emissions that I've been talking about. So that's really the hope. We've just started this work. We're very, very excited about the promise of the future of this research. All right, next slide. All right, and then to summarize the second slide, it really is looking at the carbon that's currently in the atmosphere and trying to push that carbon through plants, through microbes, and to try to capture it deeper into the soil. And by doing that, you're not only increasing the carbon in the organic matter that's present in the soil, but you're also increasing the nitrogen content. And then there's the potential to have sort of soil amendments as well as exudates that are coming out of the roots of the plants and out of the roots of the crops to be able to help sort of capture and agglomerate sort of the carbon that is exuded and captured in the soil to be able to keep it for longer and longer periods of time. And the deeper and deeper that soil goes, whether it's deeper roots, producing more recalcitrant roots, or working with those microorganisms that I talked about earlier that are non-photosynthetic that reside a meter or deeper into the soil, that's where we really want to take advantage of these microbes, these understudied microbes that we think can sort of hold the key to be able to capture higher and higher concentrations of carbon in the soil and in the deep soil. All right, next slide. All right, yeah, we can just go through all of that. Thank you. All right, so we have a couple early results here. We have results where we think we can increase the crop photosynthesis. We've engineered the carbon concentrating light and reaction pathways. That's photosystem 1, photosystem 2, and rubisco. We feel like we can engineer those systems into plants together to be able to create a better photosynthesis, an improved photosynthesis. Secondly, we think we can look at these non-photosynthetic microbes. Professor Jill Banfield, who's the director of microbiology for the IGI, has studied these soil microorganisms and has discovered an unprecedented diversity in this critical rubisco enzyme that's that carbon concentrating and carbon capture enzyme. Some say it's the most important protein in the world. Well, Jill and a professor named Dave Savage together feel like they could actually improve that rubisco molecule because there's such tremendous diversity out in nature. And then finally, we're working with Professor Pam Ronald at the UC Davis, where she's looking at crop varieties, rice varieties that have deeper and more dense roots. And there's also Professor Patrick Shee, who just joined the UC Berkeley, who thinks he can engineer the biopolymers that make up those roots to be able to make them more recalcitrant to break down, to be able to store that carbon into the soil for longer periods of time. All right, next slide. All right, the last thing I wanted to talk about before passing it over to Melinda is the prospects of using greenhouse gases and waste streams as feedstocks for biomanufacturing processes. Right now, biomanufacturing is exploding. You've probably heard of the bioeconomy. Organizations or companies like Zymergen, Ginkgo, they're just expanding exponentially. Trust me, I've seen many of my ex-DARPA colleagues find homes up at Ginkgo Bioworks in Boston recently. It's because there's so much potential in biomanufacturing to make products in a much more carbon-friendly way, as well as in a more environmentally friendly way. But one of the secrets to that manufacturing process right now is that it's run off of sugar, pure sucrose and glucose. These things obviously have to come from relatively energy dependent and carbon intensive farming practices that, as I just discussed in all the previous slides, oftentimes are not very carbon friendly farming practices. So we believe that there's a tremendous potential to directly utilize both methane and CO2 to feed into bioreactors and directly use that as a feedstock. Or alternatively, take waste, solid wastes and biomasses and have microorganisms break down those and enzymes break down those more recalcitrant waste streams to be able to use that as a stepping stone to be able to create both fuels and additives and building materials. I funded a program when I was at DARPA that was dealing with living cement. It's actually cement that has living microbes in it that harvest and capture CO2 out of the environment while the cement is curing. Right now, current cement cures and releases greenhouse gases. So wouldn't it be great to be able to expand that type of work where you're actually using it to build industrial materials or fuels and chemicals in this more environmentally and climate friendly way. So the concept is to create biomanufacturing that now consumes greenhouse gases rather than emits and relies on very carbon unfriendly farming practices. All right, next slide. And at the IGI, we've made some progress here. Dave Savage, who I mentioned earlier, has actually engineered an E. coli, which is the normal biomanufacturing bug. It's sort of like the workhorse microbe that almost all in biomanufacturing uses. Well, he's engineered this organism to actually directly use CO2 gas rather than relying on glucose and sucrose. So that's tremendously exciting. I think that there's a huge amount of potential there to be able to follow up and make that process even more efficient. Secondly, we are sort of the gene editing institute, and there are new gene editors. I mentioned these before, that it's not just CRISPR-Cas9, which was sort of the workhorse in the gene editing field, but there's a new CRISPR-Cas3, which is used to make very large deletions out of microbes. And we have a partner and a member at IGI that has created a way to be able to make these very large deletions in microbes to potentially be able to make organisms that have a very small genome that could run more efficiently in bioreactors, that could consume less carbon and be able to create and convert more of that carbon directly into the products that we want to use through that biomanufacturing process. And then finally, we have the opportunity to engineer and optimize the microbes that have naturally evolved to grow on greenhouse gases. That's really moving beyond these workhorses like E. coli and now trying to start to create new microbes that are new biomanufacturing processes. And the breakthrough that we've got here is that we've made genome scale deletions in some non-traditional microbial chassis And I think this is a really exciting area. I just attended a workshop this morning from my old office at DARPA Biological Technologies office that's looking at harvesting rare earth metals, rare earth elements, using some of these non-traditional chassis. And I think that there's real opportunity to sort of shift from very carbon intensive manufacturing to very carbon efficient manufacturing using some of these biological processes. Okay, and I think that's the end of my presentation. If we go to the next slide, I think I'm going to pass it over to my great colleague, Melinda Kliegman. Thank you, Melinda.
Thank you very much, Brad. So as the Public Impact Director at the IGI, my team works on advancing public understanding of the benefits of genome editing, and also making sure that those benefits are advanced in an equitable and ethical way. I think as many of you listen to the presentations about the research, you may have some questions about how such a powerful technology could impact society, as well as the ethics and the public perception of doing so. And these are all areas that my team investigates and tries actively to address. So today I'll talk a little bit about regulations and how those play a role here, as well as the public perception. So as Brad mentioned, there's a significant amount of research that's ongoing in the lab, but as you can imagine, that is not the end of the story. So there are many steps from getting a product from the lab into farmers' fields and then to consumers. Most organizations follow some kind of a product development pipeline. And at the very simplified level, researchers identify and develop new improved approaches or tools. So some of the new CRISPR-Cas systems like Brad was mentioning. And then they use those tools to identify new traits that are sources of things like stress tolerance, quality, or that can help with climate adaptation. And then these traits are introduced into a breeding pipeline where we can integrate them into a high performing variety or something that's locally adapted that a farmer might want to grow. And then from there, things can change a lot depending on how the product is categorized. So most of the times you'll want to do field trials to make sure that that product is acting the way that you want it to act in the field. But if your product is determined to be a GMO because you are changing the genes, then you may be required to conduct a battery of tests around product safety and put all that data together into quite a large dossier and then submit it to a regulatory agency. And that is country by country, depending on where you're trying to operate. But with some genome edited products, because they're so similar to conventionally bred products, they are deemed unregulated in some countries, and they're able to just go through the process like a conventionally bred product, where you just scale it up and distribute it according to the appropriate channels within that country. So the time and cost of all of this really depends on where you're located, and a lot of that has to do with the regulatory requirements. So since this is the Carbon Removal Law and Policy Network, we can talk a little bit about what kind of policies we're seeing globally for these new technologies. Globally, I think regulators have taken a fairly practical approach that is informed by years of experiences with transgenic or GMO plants. I'll sort of use transgenic, GMO, and GM somewhat interchangeably. But globally, most regulators do not want to have to issue new laws. It's difficult. It can take a lot of time, although some are doing so, for example, India. So what regulators have done instead is to interpret their existing laws to determine whether these new genome edited products fall within the existing definition of a GMO, or if it's excluded and can go through the pipeline as a conventionally bred product. So most of the language in the existing laws makes reference to recombinant DNA technology or a novel combination of genes as the regulatory trigger. So with genome editing, you can do things like make very targeted single point mutations, or you can make deletions of sections of genes, which silences them and makes them inactive. Or you can introduce genes from a sexually compatible relative but you can just do that in one generation instead of traditional crossbreeding, which can take many generations to get to the thing that you are really looking for. But all of those different processes, like single point mutations or deletions, they happen frequently in nature. And obviously sexual crossing can happen frequently in nature. So countries are saying that if these products are similar to something you can get by conventional crossbreeding, they're not going to regulate them under rules meant to provide oversight for biotechnology products. Of course, they're going to be subject to regular food safety laws like any product would be. And this is also practical in a sense because if it could have occurred naturally, then it's impossible to tell them apart from a naturally occurring variety. And so regulators have no real way of enforcing requirements. And I would argue that that's just not the point of regulation. If two things look the same, one shouldn't be regulated differently from the other. So as you can see from this map, most of the countries in the Americas have taken this approach. And several countries in Asia and Africa have either taken this approach or are considering similar approaches. The one outlier has been the EU, where a court case determined that genome edited products fell within their definition of a GMO as was written in the law. But earlier this year in April, the Food Safety Authority there said that their law as written is not fit for purpose, and they're going to have to go back and update the language in that law. So the countries that haven't said anything yet, most of them are just treating genome edited products as being GMOs until they can establish their laws. But a big part of the impetus of why the EU is doing what they're doing right now, I think is because they really see the benefits for climate change adaptation, especially in relation to their aggressive climate agenda and their farm to fork strategy that they're currently establishing. So how big of a difference can this regulatory burden make? So experts in the biotech industry estimates it takes about 14 years to commercialize a GM product, anywhere from 25 million to 130 million. About 5 years of that time is taken up just collecting the data for the regulatory dossiers, submitting it, and waiting for regulatory approvals. And these same experts estimate a much reduced cost and time for genome edited products at about 5 years and 10 million. I think part of it has to do with the ease of breeding, and then part of it has to do with the regulatory requirements. And so all of those estimations were made in 2018, 2019 by several different companies. So the question is, is this what we are actually seeing in practice? So in 2015, Argentinian regulators really pioneered the model of how they're going to regulate these new breeding techniques. They do it on a case-by-case approach. They review the product, determine whether it meets their definition of a GMO. If it doesn't have a novel combination of genetic material, then it's considered similar to a conventionally bred product. And so it's been 6 years, so we have some data. In 2020, the key regulators in that country published a paper summarizing what they'd found thus far. And it provides an early example of the impact of regulations on innovation. So right now on the screen, this on the top left, you see the developer type that have submitted products. The first approval was made in 1996 of a herbicide-tolerant soybean. And you can see from this graph that about 90% of the submissions were made by foreign multinational companies. About 10 institutions have submitted all of the GM products in the last 23 years in Argentina. And if we compare that to genome edited applications by developers, we see a much more even distribution of who's submitting applications. There have already been 13 different developers, dominated by local companies and public research institutions. And this is a comparison of 23 years to about a 3-year period. And the same thing, if we drop down and look by product type, we can already see a much wider variety of products, even in that shortened 3-year time period. So what this highlights to me is things that people have been saying for a long time without a lot of concrete evidence. It shows that the cost of regulation can really shrink the number of players that are able to operate. And then even for those large multinational companies, they're really limited in what they can develop because they only focus on crops that can be the most profitable because the regulatory burden is so high. And I think there's a lot of calls by civil society organizations for increased regulations aimed explicitly at limiting the activity of multinational companies. The conversation has shifted away from safety to monocultures or the overuse of particular chemicals. But I think it actually has the opposite effect where the additional regulation makes it so expensive and difficult that it's only the largest players working on a tiny fraction of crops, traits that can have access. And that actually ends up leading to consolidation and monocultures. And if you lower the regulatory barrier, you see a widening field of small companies, local and public research institutions like our own, and you see a wider range of product types. So you might see things that are more consumer facing or climate change oriented, instead of solely focusing on farmer profits. So I think that's all really interesting, and I'm curious to see how it evolves in the future. So then the last thing I'll talk about that might be of interest is just the public perception. I think we can't really ignore this since it's part of the negative feedback loop, at least around GM products. And it leads to this high regulatory requirements and potentially consumer rejection in some places. So it's a big concern. And as scientists, we want to appreciate it. We want to address it where possible before products reach the market. So to give you an example, this paper by Yang and Hobbs in 2020 published a study on the perceptions of genetic modification and genome editing. And people were asked to rate from 1 to 6 whether they thought these products were natural, safe, and ethical. And on this graph, on the y-axis, we have a rating scale with 1 being not at all, safe, natural, ethical, and 6 being completely. The colors are, the darkest blue for crossbred plants, which I think is interesting that like traditional crossbreeding didn't get closer to a 6. And then the light blue is for mutagenesis, where plants are irradiated with chemicals or radiation to generate random mutations that breeders can then select for. And that's rated quite lowly across the board, although it's been used for a long time and is considered a conventional breeding technique that doesn't require further examination by regulatory agencies. And then the horizontal hash lines are GM or transgenics, and the hash lines at an angle are genome edits, which get somewhat of an intermediary score here. And then if we look across the published literature, there hasn't been that much to date because genome editing is still fairly new. There are publications that look at the attitudes, perceptions. Some have measured the willingness to buy and at what cost. And some studies have looked across multiple countries. So it kind of highlights the perception differences across countries. So I grossly oversimplified this to compare them roughly, but the 2 plus signs are a positive perception of the breeding technique. A positive and a plus and minus sign is sort of a mixed perception or something intermediary from the highest and lowest scores. And then the 2 negative signs indicate that consumers have a more negative association. I think overall, on the most basic level, the research shows that consumers find crossbreeding to be the safest, and they're more open to genome edited products than GMOs. And then, of course, there's the interesting part of the mutagenesis that consumers rate even lower than GM. But one of these studies found that when consumers found out they'd already been eating products of mutagenesis for many years, it raised their perception of it. And it also mattered to consumers who developed the products. If it was a government or public sector developer, they had more trust in that, and that was preferred. So I think it is important for us as scientists to think about the research and sit on it a little bit. If we do things the way we've always done it. So for instance, some of these studies from France, the consumers say, we don't want anything new. We don't want any changes at all. If we go that route, then there's no innovation, there's no progress. And I think in the long term, that's suboptimal for society. But on the flip side, we want to bring the public along in the process and make sure that the societal values are integrated into our research. So a big part of that is having dialogue with stakeholders to really understand the nuances of their needs and perceptions. The IGI has conducted outreach. We recently held a climate workshop in April of this year where we invited environmental nonprofits, farmers, civil society organizations, governments, and international organizations to start an early dialogue with us where we can share our approach before we're too far down the road, and get honest feedback on the best ways for us to proceed. We would like to innovate, and we are innovating and lending our expertise towards solving the climate crisis, but we also want to honor our shared values around environmental impacts. And we believe that these dialogues go a really long way in improving the public perception, since we want to act in a fashion that actually is worthy of trust. And as an outcome of this meeting, we also spoke with the US Department of Agriculture regarding their own climate agenda. And they want to empower farmers to use tools at their disposal to reduce their carbon footprint. And this is definitely some tools that could aid in that. But we're all kind of trying to figure out how to make sure we can measure the carbon that is stored and for how long it's stored. I think the policymakers at the USDA signal that that is something that they're very much working on right now. So with that, I will stop there and thank you all for your attention, and I guess turn it back over to you, Will, for questions.
Thank you very much, Melinda, and thank you, Brad. And I will turn to the questions now. Brad's been kind enough to answer most of the questions in the interim, but given the fact that many may not have seen those answers, and I can never remember if they're captured in our recorded version, I'm going to try to work through some of them. So I apologize, Brad, if you have to.
No, absolutely, Will. And there might be follow-ups too from my answers. So I'd love to have a discussion for sure.
That sounds great. So the first couple of questions are focused on the issues of permanence and recalcitrance, both of which were discussed a bit already. So the first question is, will continued large-scale wildfires negate the potential to store significant carbon in new growth biomass?
Yeah, and my answer to that really agrees. I mean, this is a critical question, and I think that it goes towards a couple of the subsequent questions that were listed there as well. Is that permanency, that recalcitrant nature of the biomass, and you don't have to look any further than the wildfires that are ravaging the West Coast right now. It's a big question. Now, if you think about that in terms of agricultural, because most of our work that we're going to be pursuing is in agricultural lands and sort of increasing that agricultural biomass. So the problem with agricultural biomass isn't probably as much a wildfire as it is with sort of the leftover biomass, the waste biomass. That's the roots, that's the leaves, that's the part of the plants that aren't really eaten or consumed for nutrition. That's where we think we can actually come and step in to be able to incorporate that biomass more completely into forms of carbon that persist for longer periods of time in the soil. The added benefit of that is that it should be recalcitrant to things like fires and tilling and like these practices that oftentimes result in reductions in emissions of carbon. And my subsequent answers to a lot of the questions after this sort of go into a little bit more detail about how we hope to try to do this. So Will, maybe I'll just go a little bit through a couple of the approaches that we're thinking about. So
that'd be great.
Look, there's been some well-publicized work by Joanne Corey down at the Salk Institute that's looking at trying to create more recalcitrant roots. Thumbs up, like that is fantastic. That's the right approach. We want to try to add on to that work a little bit for some of our work. We do believe that just one type of biopolymer, I think they're pursuing suberin. Our approach might go a little bit beyond suberin, that there's some reports that suberin can, regardless of how it's engineered, could still be broken down by sort of the complex microbial kind of inhabitants of soil. So we want to try to create sort of a mesh network. Think of like a patchwork quilt of biopolymers that I think might be able to interlock with each other a little bit to really create a much more recalcitrant form of biomass, at least subterranean root biomass. And so this is work that's being proposed by and started by Patrick Shih at UC Berkeley. And he's looking at things like sporopollenin, which has been shown through fossil records to exist in nature for hundreds of thousands to millions of years. Trying to combine that with some of the other natural polymers like suberin or other to create these networks of roots. So that's one thing, is to make more recalcitrant roots. But in reality, I don't want to put all the eggs in that basket, because if we create roots that don't biodegrade, like that kind of sounds like plastic to me a little bit. So I really want to try to focus a little bit on root exudates as well. And root exudates are essentially the metabolites, the sort of the carbon that comes out of the root systems that gets deposited into soil. So you have all of the above ground biomass. That's the fruit and the cereals and the grains and things, as well as the leaves and the stem of the biomass. But then you have all of this carbon that gets deposited into the soil. That essentially feeds the microbial biomass that's in the soil. Right now, if it's just left uncontrolled, the natural sort of circumstances of carbon and the carbon evolution in that soil, oftentimes that carbon will be released as CO2 or methane into the environment. We think as we characterize all of the microbes that are present in soil, we can start to understand the networks and the interconnectivity between those really complicated interactions between those soil microbes. There's literally 1 billion microbes per cubic centimeter of soil. So we're going to characterize all of those microbes, do it at a genome scale, so we understand the mechanisms, the connections between the different carbon pathways and the carbon metabolites that are there. That will inform how we could then edit crops to be able to sort of talk with those microbes through the funneling of these metabolites and exudates out of the roots. And we think we can upregulate the microbes and increase the population of the microbes that better fix carbon, that increase the soil aggregation, that has been shown to be able to extend the period of time that carbon is stored in the soil. So that's a little bit of a nutshell of some of the things that I was saying, but please do look at some of my answers where I typed out in a little bit more detail some of the things that we're doing. But we are not looking at just spraying microbes onto the soil. That's not what we want to try to do. We want to be able to increase the population of beneficial microbes in the soil, to be able to increase aggregation, increase organic mineral associations, and then be able to increase the root and the recalcitrantness of the roots and sort of the microbial networks that are in soil. Okay, so those pre-existing things that are there, we want to be able to sort of increase the populations of those, not have to spray.
All right, thank you for that, Brad. We have a lot of other science-oriented questions, but I want to make sure we get a bit to on the regulatory side too. And I had a question for you, Melinda, related to your interaction with USDA and thinking about the role of genomics. As you acknowledged, there's a lot of serious questions about being able to both monitor and quantify soil sequestration associated with various practices. Has your institute or the USDA indicated that there may be some unique challenges in measuring soil sequestration associated with the techniques that you're contemplating using?
Yeah, I mean, from our discussion with the USDA, it seemed like they were very much in the process of trying to figure out a path forward. It did not seem like they quite had an answer to it yet. I think one of the things that the secretary said was that he wanted farmers to think about profitability instead of just yields. So think about the whole context, including potentially subsidies for sequestering carbon on their farms. But beyond that, we haven't seen anything from them so far on exactly how they would move that forward. And then maybe I could throw it over to Brad. I don't know what we're doing in terms of thinking about measuring carbon sequestration in the soil.
Yeah, and that was one of the questions that was in
one.
the chat too, Melinda. Look, I mentioned one of the professors and scientists that we're working with, Jennifer Petridge. She has a joint appointment between UC Berkeley and Lawrence Livermore National Lab. She works closely with another professor named Mary Firestone, and their work, and they've published some of this, is specifically looking at sort of the carbon residence times in soil. They've done some really preeminent work in sugarcane and looking at sugarcane roots and root structures, as well as root exudates, where they're looking at what types of structures and associations and agglomerations in soil result in longer times of carbon storage. And additionally, also the depth into the soil makes a significant difference in how long the carbon resides in the soil. The deeper you are in the soil, the longer that carbon is going to be able to persist. So those are the individuals that really are going to make those measurements. And oftentimes, what they do is create sort of an ecosystem in the laboratory where they can actually do isotope tracing. And those isotopes then are the sort of the half-life of those isotopes can be used to try to understand sort of how long these agglomerations and sort of aggregations in the soil could potentially persist into the future.
Yeah, but I think, Will, on a broader scale, I think this is something that the field as a whole is struggling with to do on any kind of a large scale. Like, of course, you can go into one particular farm and test, but if you're going to deploy something across a huge scale and potentially provide subsidies for it, then it starts to just become very challenging.
Yeah, and I do think that NIST may be able to play a role here. This is the National Institute of Standards and Technology. This seems like a role that NIST might be able to play, maybe to cross over and work with the USDA a little bit, or the DOE, to try to figure out measurement strategies and sort of quantitation and reproducibility of that. I think there might be an opportunity, Will, for you and your organization to reach out to some people at NIST. There may be some interest there.
Yeah, I think that's an interesting question. I think as is the case with a lot of these things, there's a lot of siloing still, right? And a good thing to look at. All right, thank you very much for that, both of you. The next question is more economic in orientation. It says the US government invested 100 million in failed biomass to fuel efforts in the 70s. Again, in the 2000s, 500 million in failed biomass to fuel research was funded. In the current effort at using biological processes to utilize methane and CO2, are they doomed to be another technical success but economic failure?
Yeah, that's a great question. And again, these are great questions. I couldn't agree more. Look, having been a guy that was responsible for a $300 million a year budget at DARPA, I am highly sensitive to the wasting of government funding. And look, we need better strategies for how to do an economically viable biomanufacturing process. And so that's when we devised this strategy, that's the very first thing that came to our mind, because we want to utilize waste. What better to start with than waste? And if you can't make something economically viable out of a waste product, whether that's a gas waste product or a waste stream of some sort, brewery waste, gas waste coming off of a steel factory, you name it. There's a bunch of different waste streams that could potentially be used. That's the hope, is that if you can make that viable, if you can turn that into a biomanufacturing process, now you don't have to rely on pure forms of carbon like sugar and sucrose and glucose. And at the same time, you're using a feedstock that in most cases, companies and the producers of that waste will literally either pay you or it will be free to you to start with. And so that's the hope, is that it potentially could be an economically viable source. But let me point out that a lot of what we're talking about is tremendously difficult. The solubility of something like methane is tremendously low. So how do you get that methane to make it biologically available to a microbe? That's an unsolved problem. That's something that's going to require basic research. Using new microbes that are not E. coli or yeast that are naturally evolved to be able to utilize things like CO2 directly. How do you make those at scale and be able to make those efficiently produce the chemicals and manufacturing products that you need? These are things that we're currently working on, but it is tremendously difficult.
Thank you for that. This is another question that's related to the economics. Will credits to farmers be required as incentives to apply the techniques, or will these modified crops and microbes be economically viable through things such as producing higher yields and lower fertilizer costs, and thus not require subsidies?
I'm going to toss that one
I
over to
mean,
Melinda.
I think, you know, the hope and the plan is that you will have higher yields, is that the soil will be more fertile. And that in and of itself should incentivize farmers to adopt these practices. But again, you know, farmers can be a little bit risk averse. I mean, their entire livelihoods depend on having a good yield. And so trying out something so new, which may also have some more longer term benefits that might not be realized in just one cropping season, you know, it could require incentives at least at the beginning. And it seems like at least our administration, and I'm fairly sure the administration in the EU is really interested in anything that would help reduce the carbon footprint of agriculture, because there's just not a lot of options available right now. So I mean, there are a lot of subsidies out there for other things that are not as sustainable or climate friendly. So I think adding an incentive on top of it is never a bad thing if you're looking at increasing sustainability.
Yeah, I had a kind of a follow-up question related to that. You showed us all the data, Melinda, in terms of public attitudes toward various approaches. Have there been surveys of farmers specifically about these approaches, and what did the surveys yield?
Mm-hmm. I mean, so to just back up and put it in context. So, you know, about 90% of the, or more of the corn, soybean, you know, sugar beets in the US, grown in the US, are all GMOs. So farmers have already heavily adopted new technologies and are open to embracing them. I think it's more on the consumer side where we're seeing some, you know, rejection or pushback. So from the farmers that we've interacted with, you know, they are very open to trying new technologies and new techniques, but you know, they just want to make sure that what they're going to get on the back end is, again, going to be economically viable. And some of them are also very concerned about sustainability and climate change.
Okay, thank you for that. There was another question, must have gone to the answered section, but asked about your focus on soils and agriculture, and why not in the context of forests.
Yeah, I saw that one and tried to knock that out, Will, because it is really an interesting question. Melinda, I'd love your thoughts on this. I basically said, look, we've had these discussions internally, and it's absolutely a bandwidth issue inside the Institute. We basically had to choose whether we were going to go. We assembled Pam Ronald, Brian Statskowitz, these members of the National Academy of Science, and basically said, you know, what are we going to do? How can we make the biggest impact with the limited resources that we have? And we really chose things like rice that have a significant carbon footprint and are a significant climate problem. And we decided to go after that and soil as sort of our first chunk, but it's absolutely on our radar. We do have some thoughts and ideas there, but we just don't. But Melinda, I was going to pass it over to you for sort of the regulatory perspective of that as well.
Yeah, I mean, there's a more practical reason, right? So agricultural lands are already disturbed. So, you know, if you're going to make some kind of change there, you don't have to be as concerned about the ecological interactions with other organisms. So that's sort of one context. They are also sort of, at least with GMOs, right? They're also somewhat cut off from those interactions with the nearby ecology. So there is a pathway forward, an established system for testing out new GM products. Regulators have experiences with them. And there's a way to sort of confine your field trial so that you are only looking at what's happening within that system. And so it's a good way to start. The agricultural systems are a good way to start, good way to kind of test out your different theories, make sure things are working as you imagine. It's a good way to kind of liaise with the regulators as a first step. And then from there, you can kind of go into these more complex, undisturbed ecological systems. One good example is, you know, the GMO chestnut. that has recently been approved by regulators. I mean, I think it took over 10 years for that to go through the regulatory process, as opposed to, you know, another corn or soybean that's going into an agricultural system that regulators have a lot of experience with. So it's a little bit practical too on the regulatory side.
Thank you for that. I think we have time for one more question in here. The question is, if these crops are not GMOs, could they be grown by organic farmers and labeled as organic? And could they be integrated into farms using regenerative agriculture techniques?
Yeah, so that's still a big open question that we actually asked the USDA as well. So there's, you know, a few different agencies that are working on GMOs. There's APHIS, which is the biotech regulator. They have said, you know, these products are not GMOs. Then there's the Agricultural Marketing Service, which handles the GMO labels. And they have also said, you know, you're not going to be required to label this as bioengineered if you're genome edited. And then there's the Organic Standards organization, which hasn't really said anything yet. There's an Organic Standards Board that makes recommendations to the Organics Agency, and they have recommended that genome edited products not be included in the Organic Standard. But then I think internally in the agency, there's this tension because you don't want to have different parts of the same agency making somewhat conflicting determinations. So we don't really know what the final answer is going to be yet. But again, I would argue that if the goal of these products is really focused on sustainability, then I would say that aligns very well with the values around organics. If you're really reducing pesticide use, reducing herbicide use, for instance, then I think that aligns well with the values of what the Organic Standard is trying to do. So open question, I think it should be included, but right now, from the regulatory policy perspective, not sure yet.
Thank you very much for that. We have just enough time for any final thoughts either one of you might have in this context.
Well, I really appreciate the thoughtful questions. This has been a tremendously interesting forum, and the active questions is just absolutely phenomenal. And this is why I came to the IGI, was to be able to have these types of discussions. So I really, I salute those in attendance as well as your institute, Will. You guys are doing a great job. And again, I posted my email, but it's bringeisen, just the letter B, and then ringeisen at berkeley.edu. I'm happy to continue any of these conversations with people that are interested. This is what we need to do as a society, is to have these conversations. We don't have time. We ran out of time about probably 10 or 20 years ago. So we need to do this, not tomorrow, but we need to do it today and yesterday. So that's, I'm really excited about the prospects of the research that we're doing, but having these kind of forums and discussions and people asking the types of questions that they asked are exactly the right thing to be doing right now. So I really think it's been a great session.
Okay. Anything else, Melinda?
No, that's it. Thank you all. I just want to reiterate what Brad was saying. Like, these questions are just really amazing. And I think we need to go back and sit on some of them for a little while. It's great to talk to smart people like your viewers and listeners who are well steeped in this field and get a little bit of a different perspective. You know, we work a lot with molecular biologists, and so it's good to interact with people on the other side of things as well.
It's a pretty cool community. So yeah, we hope you will. All right, so we are at the top of the hour. And so with that, I'd like to thank our two speakers today, Brad and Melinda, and of course, all of our participants. And I hope to see you again soon. If you're not on the Institute's mailing list, please do so. And we will apprise you of future events that may be relevant. And with that, I will close this session. Thank you, everyone.
Thank you.
Thank you.